That's working, right? Okay. All right. Well, morning, everyone. So it's my pleasure this morning to welcome Dan Simionescu and also Aggie Simionescu to the department. So Dan's going to speak to us today, but I'd just start off by saying that the two of them are stalwarts in their Department of Biomedical Engineering and at Clemson. They run independent laboratories, but they very much work together, and they're an impressive and productive team. So Dan is Harriet and Jerry Dempsey Professor, an endowed chair in the Biomedical Engineering Department. And his work is very much focused on artificial heart valves, vascular grafts, myocardial patches. He's a great basic scientist, but he has a sort of real translational bent as well. Very much interested in seeing the products of his research have an impact on medicine and how it's practiced. He's an elected fellow of the American Institute of Medical and Biological Engineering, and also we were just chatting a second ago. He received an honorary degree for his contributions to science just in 2017 from his old school in Romania. I'm guessing it's the highest honor that they confer upon a graduate of the schools. So that's pretty impressive. He's an editor of a number of textbooks, including one on vascular genesis and angiogenesis, and the editor-in-chief of Challenges in Regenerative Medicine. And I noticed, looking over his CV, he's got a bunch of patents, and they're pretty impressive. And really what makes them impressive is about half of them are licensed to different companies. So, it's one thing to produce a patent. It's another thing for an actual company to become interested in that patent and practice it as part of their business strategy. So, I was duly impressed by that. So, he's also very well funded. He has a large R01 size grant. It's a collaborative grant with his old school in Romania, but he's also funded by the NIH and the American Heart Association. And I'll just mention that Aggie just received the good news that she received an R01 award on her work on diabetes and vascular problems. The other thing I'll mention about both of them is that they're part of sort of distinguished scientific lineages. And I know Daniel probably hates me mentioning this, but his dad was a very famous scientist. He worked with George Palade back in the 1970s, and Palade, of course, won the Nobel Prize for his work. But Nicolae Simionescu made some major contributions to that work that led to that Nobel Prize. And in fact, he returned to his native Romania and set up a very well-regarded research institute there. He numbers around 600 publications, just a very impressive career. And I think the two of them are very much, and also his mother as well. It was his longtime collaborator. So just a very impressive scientific lineage. So I'm looking forward to Daniel's talk today on the Valve Regeneration Project, and I'd welcome you up to the podium here, Daniel.
Thank you. Hello, everyone. Thank you so much, Rob, for the introduction, and also I truly appreciate the invitation to come here. We have been meeting on and off in different occasions with Dr. Gurdi, and the last one was maybe 2 years ago in Spartanburg, if I remember well, where he gave an amazing lecture on his work, and we got a chance to talk a little bit about that. I also appreciate the opportunity to be here and talk to you guys, to some of you. We talked to a couple of you last night and this morning as well, and I realized that the work that I'm going to present it's more of a tissue engineering or biomaterials kind of technology work. Maybe something that could be interesting for you. And as Rob said, in the last 10 years or so, I have been keeping my focus on translational. It's definitely very, very challenging, and I'll talk about some challenges towards the end, but I think it could be very rewarding to accelerate the pace towards testing, developing devices and testing devices, so that maybe one day in your lifetime or maybe later, it could help a patient. So the VALVE-REGEN project has this acronym because it is a collaboration between Clemson University Bioengineering, shown here. Everybody recognizes the tiger paw, and the University of Medicine and Pharmacy and Science and Technology of Târgu Mureș in Romania. And I'll explain to you what this collaboration is all about. The main focus of our research is the aortic valve. Everybody knows about it. I'm always liking to use this quote from Dr. Nishimura: is the most frequently diseased, replaced, and researched valve out of the 4 valves that we have. And also, from the bioengineering point of view, you have to consider that this is the most mechanically stressed tissue in the body. If you look at the images here, which shows you an aortic valve functioning every second, pretty much. It has to function continuously. There's no other tissue in the body that has to bend and stretch and open and close every second. If you do the math, it's about 3.2 billion times per an average lifetime. So one question that we have been asking ourselves and continuously looking for answers is, what is the secret? Why do some people have perfectly well-functioning valves throughout their lifetime? What is the secret to valve durability? And looking at different angles of understanding valve durability, looking at diseased valves, and also looking at design principles that we have been using to develop artificial heart valves, we kind of decided that we're going to look at 2 unique structures. One is the actual 3-dimensional structure, and I'll talk to you about that in a second. What is the matrix distribution of different components and microdomains, and also the cells? Why are these cells so specific, and how are they capable of maintaining the extracellular matrix under these conditions? So if you start with the structure, this is a typical aortic root from porcine sources. And we decided we're going to look at the whole aortic root as a dynamic functional unit and not only the cusps that open and close all the time. Actually, the root opens, dilates, and then recoils during the functional cycle of the heart. So this is a view from above, a view from the bottom. If you cut it open, you see the 3 cusps here, the 3 little pockets that constitutes the aortic valve. Now, this 3-dimensional dynamic structure is important because as the valve opens and closes, there is a very important stress distribution on the leaflets, as you see here. This is a simulation in the lab. And if you look at the cross-sectional structure of the valve, this is the sinus, the annulus. So this would be a cross-section about here through the bottom, through the middle of the valve. This is the thin leaflet of the valve. You notice the distribution, the microdomain distribution of collagen, which is blue here, elastin, which is brown, you can barely see it, and the cells in different structures. So there is a particular structure and 3-dimensional distribution of matrix that accounts for this durability. If you look at the cells, this is one of the cusps that is cut out, looks like this. If you look at the cells, you recognize that the structure of the valve actually has 3 layers: fibrosa, spongiosa, ventricularis. the cells are distributed through these structures. And also, there is a preferential distribution of collagen, which is yellow here in this pentachrome stain. GAGs are distributed mostly in the spongiosa and elastin mostly in the ventricularis, in the layer that faces the ventricle. The cells, even though we have done a lot of research on them, we still don't have a name for them. They're continuously called valvular interstitial cells or valvular fibroblasts. They do behave like fibroblasts, but they have very specific properties that differentiate them from fibroblasts. What do they do? We believe that the cells, as a response to micromechanic stimuli, think of the tissue being, the valve being open and closed, the tissue being bent and recoiled, those stimuli allow the cells to react to that and maintain ECM homeostasis. And I will show you a little bit of data of how we think that is actually happening. And probably having healthy valvular interstitial cells ensure durability of the valve in the heart. Unfortunately, valves are affected by pathology. You probably know about stenotic aortic valvular disease. This is defined as calcification, mostly in the cusp structures. This is prevalent in 3 to 4% of population. There is an association with age. There is an association with genetics, diabetes, atherosclerosis. These are just the symptoms. More importantly, aortic valve stenosis may lead to heart failure, and this is very, very significant. To date, we do not have drug therapy for valve degeneration. So understanding the mechanism of pathology is a very important chapter of this field. And practically, surgeons now have some artificial devices that they can use to replace the valve. This is the treatment for valve stenosis, surgical replacement. We also have candidates for new types of valves. These are the congenital defects, such as bicuspid valves. So just a quick summary of what is out there on the market. We have homograft. This is applicable to all ages. These are pretty much human valves, cryopreserved. They are quite durable, but they come in limited supply. They look like this. Mechanical valves, which look like this, are made of carbon. They're very thrombogenic, so the patients have to survive an entire life of anticoagulation medication. Tissue valves are made of glutaraldehyde fixed animal tissues, such as pericardium or actual aortic valves. Unfortunately, these degenerate within 10 to 15 years by calcification, and polymeric valve, which is only in experimental stages. I wanted to highlight the autograft alternative, which is not very common because it's technically very difficult. It's called the Ross operation, highlighted here, where the surgeon will take a healthy pulmonary valve and replace a diseased aortic valve within the same patient. Now, the main limitation for that is that you would need a homograft or some kind of a new valve to be placed in the pulmonary position. But these are the most durable valves from clinical studies. And so overall, there is a clinical need for a new type of living functional valve, mostly for young and adult patients. The prosthetic heart valves, the mechanical, the tissue valves do not function well in these patient categories. The prerequisites are at least to have proper hemodynamics, sturdy structures, and we believe that these valves should contain living cells that would maintain the extracellular matrix. And of course, try to reduce the risks for thrombosis, degeneration, calcification. For children that have not yet completed somatic growth, the device should be able to grow with the growth of the heart. So our research goal in the last 10 years or more is to generate implantable living valves, combining structure, the three-dimensional structure, and the cells. And I'm using a quote by Sir Magdi Yaqub, one of the more famous heart surgeons from the UK, which said that the clinical relevance of a living valve substitute has been confirmed by the Ross procedure, where you take a valve, you pretty much transplant it into a new position, and implanting a living valve will significantly improve clinical outcome. So encouraged by this statement, which was made 5 years ago, we continue to work on heart valve tissue regeneration. So just to take a quick break, the take-home message number 1 is that valves are intricate structures. They have specific hemodynamics. There's a complex cell and matrix biology. We do not understand pathogenesis yet, and the replacements that are out there have some limitations, but there is still a significant clinical need. So we are considering these aspects when designing a regenerative approach. So our approach involves using a 3-dimensional structure, which is derived from a decellularized aortic and pulmonary valves from ovine and porcine sources. We seed these with cells. We now use autologous adult stem cells from adipose tissue. We differentiate these stem cells into fibroblasts and endothelial cells, seed those cells in the appropriate anatomical areas, and prepare them for implantation using dynamic conditioning in heart valve bioreactors. So our working hypothesis is that acellular tissues could serve as cell niches or scaffold. So you start with the target tissue, let's say the valve. We remove all the cells this way using detergents and enzymes. This way we remove all the antigens and generate a niche that could be used later for cell seeding. And after mechanical conditioning, we have a regenerated target tissue. And we have applied this principle to many other tissues as well. So a simple recipe for a tissue-engineered aortic heart valve will be to have a valve matrix or a scaffold, about 10 million stem cells from the patient, 1 bioreactor, and then you preheat the oven, prepare the matrix, insert the cell into the matrix, blend, bake, and serve. And we'll see if that actually works out. One question that we have had numerous times when we submitted grant proposals and papers is it safe to use acellular tissues? I mean, we take tissues from pigs or sheep. We remove all the cells. Are those antigenic? And the answer is yes, it is safe. This is an example from a paper by Steve Badilak, which has shown that more than 1.5 million patients have been already implanted with matrix-based acellular tissues from human, bovine, porcine, equine sources. All of these have been FDA approved. And you can see the different tissues here, either naturally or cross-linked. And there was no sign of rejection or other side effects. The main reason why this is happening, we believe that collagen and elastin have been preserved throughout evolution, so they are really not antigenic when used in human patients. So we designed a translational scenario that we wanted to test in different steps the validity of the hypothesis. So we start with the patient, which would be sheep. We collect fat. We collect adipose tissues. We isolate stem cells. We now differentiate these stem cells. We seed and condition these stem cell seeded scaffolds, and then prepare the implant and implant them back into the same patient as autologous implants. In step 1, working with the stem cells, we have been working with adipose-derived stem cells and differentiate them into endothelial cells and fibroblasts separately. And here's one example of doing endothelial cell differentiation. We have grown cells on gelatin-coated plates, incubated with endothelial cell growth supplements, and exposed them to shear using an orbital shaker. And thus far, the results show that there is a tendency to commit towards an endothelial cell lineage by showing positive uptake of LDL, positive immunofluorescence for CD31, von Willebrand factor, eNOS, and so on. We're still working on other properties. So to summarize what we have done thus far, we believe that growing on gelatin, exposing to the supplement and shear, we can actually generate some kind of commitment towards endothelial cell lineage. For fibroblastic differentiation or valvular interstitial cell differentiation, we have incubated adipose-derived stem cells with TGF-beta1, low levels, and found out that this particular method allows for expression of some of the markers. Truly, fibroblasts don't have one individual markers, but a group of markers. Interestingly enough, these cells also make collagen type 1, glycosaminoglycans, MMPs, and TIMPs. So it's possible, but by using these arrays, we could highlight their ability to modulate or remodel the extracellular matrix. So overall, we believe that this method will help us commit adipose-derived stem cells to differentiate towards the fibroblast phenotype or lineage. So the take-home message number 2 is that it is possible to use adipose-derived stem cells towards using as cell sources for valve regeneration. These cells are easily obtained by liposuction or lipectomy. We have obtained it from human patients using a collaborator or from the experimental animals using simple lipectomy. Working on the decellularization phase of this project, we realized from the get-go that this type of tissue being made of different tissue types, the aorta being an elastic artery, the cusp being a collagenous, and so on, just simply using immersion decellularization did not work. So we had to design a system. This is one of the patents that was issued and licensed. We designed a system, first of all, for mounting the valve in such a way as not damaging the tissue at all and not damaging the cusps. And these valves, once mounted in the system, are put into a little bath, and a pump will push the detergents, nucleases, and other chemicals through the system, something that looks like this.
Question? When you decellularize the system and you're left with the kind of matrix basic structure,
Yes.
and then you repopulate it, does the basic structure stay the same, or is it somewhat dependent on the interaction with the cellular matrix? So if you were to do a very quantitative, which you've clearly done, structural analysis of that before and after decellularizing and recellularizing, are there any subtle changes in the basic structure?
I'll show you some data regarding after decellularization and before. So the matrix is maintained very well. After seeding cells into them, we don't know yet. We don't know yet, but we will definitely look at that. We are at that stage where seeding cells into the scaffolds, and we started implanting them into animals. So that's a very good question. Thank you for that. So of course, to provide the answer, we looked at what happens after decellularization. This is a little bit complicated, but very quickly we have looked at the aorta, the sinus, the cusp, and the muscle, the different areas in the aortic root. And this is the immersion results. Sometimes we find cells within these tissues. 16 days of decellularization in that system, in the decell system, actually works really well, removes all the nuclei, maintains all the extracellular matrix components, the collagen, the elastin. We do lose gags, and essentially we were a little bit surprised, but then we realized gags are very water soluble, so it's very difficult to keep them in the tissue. And we also look for the amounts of DNA as an indicator of presence of cells. And here you have fresh tissue in yellow and decellularized tissue in each area of the anatomical three-dimensional structure lacking any DNA. So based on this, we concluded that it's fully decellularized. We also performed extensive biomechanical testing on the cusp, the sinus, and the aorta. We also did bending analysis using a little device developed by our collaborator at Mississippi State University. Overall, we found out that after decellularization, mechanical properties were pretty much preserved, very minor changes. Of course, for a clinician, it's very important to see that the decellularized root still functions properly, and you see it here in the lower video. We have done some quantification of opening and closing characteristics shown here in this graph and calculated geometrical orifice area. And pretty much it looks like decellularization, the way we do it, preserves aortic root hemodynamics. So encouraged by this, we pursued the next steps in the translational scenario for regeneration. So the final product that we have right now is fully acellular in all the areas of the tissue. You can recognize the change of color to pretty much snowy white collagen color. It's very porous, as you see here, probably good for cell seeding. We have preserved the matrix. We lost the gags. Mechanical properties and hemodynamics were preserved. So this kind of takes us into the next step, seeding and conditioning. And the work plan was as followed. We decided to plan it a little bit better. So we started with interstitial seeding, where we aim to put fibroblasts in the cusps and in the lower portion of the aortic root. We also would work with small muscle cells, but I don't have data on that yet. In the second step, we will optimize luminal and adventitial or external seeding of endothelial cells into the lumen and fibroblast into the adventitia. And finally, after all these procedures are put together and optimized, we will recellularize the entire decelled aortic root and have something that looks like this with the right cells in the right position, ready for implantation. So I'm going to show you some experiments that we have done preliminarily. This is just injection seeding. So this is internal injection of fibroblasts into the cusp structures. We submit the valves to a rotator system just to keep the fluid moving and have some very, very slow opening of the valves for mechanical adaptation. And then we submit these seeded valves to bioreactor conditioning, which I'll explain in a second. Overall, we got okay results, but a lot of cells have disappeared, have died. Now we know why. We understand what was wrong with this particular experiment. And one of the reasons is that cells require some kind of a matrix when being injected into tissues. So the matrix that we considered was fibrin. And we found out that if you inject cells in fibrin gel, as you see here, we added a blue dye so that we could follow the injection sites. Once you inject cells in fibrin matrix, recellularization is much, much improved. We had much better cell numbers. You can see it here on live dead staining and after bioreactor conditioning. Fibrin gel degrades after a few days, so it serves as a very good temporary scaffold for internal recellularization. Recently, we have developed a new device, which is patent pending now. We coined it cytoseeder. This is based on a drum which contains the cells and a roller which is adapted with 350 microneedles on the drum. So you can load your cells into the drum, and using a simple pipette 8 kind of system, you can roll tissues and hopefully seed more cells than by doing manual injection. So if you do 10 rolls, you get 3,500 injections. And one example of how we're doing this on the valve, you take the decellularized valve, you can flip it inside out like a sock, exposing the cusps here with asterisk, and then you can roll the cusp with this device, and then flip the aortic root back and roll the outside for the adventitial. So this is in the works right now. For luminal re-cellularization with endothelial cells, it works pretty well per se without any help. The cells like the internal surface of the acellular valves. And you can see some live dead staining here of the seeded valves and SEM as well. Sometimes you see areas which are not fully covered, so this means we have to implement repetitive seeding, probably 3 or 4 times until we cover the majority of it. For adventitial seeding with fibroblasts, we designed a little system here that was 3D printed, and using this device, you can mount the valve as it's already inserted into the metallic ring, and you see the seeding here using a double-barrel syringe. One has the cell, the other has the fibrin gel, and after seeding, it was conditioned in the rotator. The results look pretty good on a live dead staining under a stereo microscope, and on trichrome stain, you see that the cells with the fibrin gel covered the adventitia pretty well. All these valves will essentially go, after seeding, will go into the Harvard bioreactor. This is a separate project that we started almost 10 years ago. And now we have a system in the lab that looks like this. This is an aortic valve holder. This would be the chamber. We have a pneumatically driven pump that pushes cell culture media through the valve, and these are reservoirs that mimic the entire human vasculature. It's all computer controlled, and we have good control over pressures, flow, and of course temperature, since the system fits very well into a simple cell culture incubator.
So Dan, the flow of the fluid is the thing that drives the opening and closing of the valve?
Yes,
yeah.
yes. So there is a membrane here that just pushes up and down the fluid. Yeah.
I was reading Da Vinci's biography by, and he described his speculations of how valve flaps work due to vortices flow beneath the valve flaps. Is that incorporated into
They
this?
close because of the vortices.
Yes.
Yes. So there's a change in there's a change in pressure. When the ventricle contracts, it pushes fluid, and it closes the mitral valve and opens the aortic valve.
Right.
And this is what we're mimicking here. This membrane pushes up exactly like the ventricle would contract and push the ejection fraction. When it closes, there are vortices which kind of swirl around in the sinus, and that helps with the closure of the valve. The difference in pressure also does that, but the vortices do that. Yeah, I'm a big fan of his as well, and I have the same diagrams that you have in your office, right, with his ideas of how the valve works, and it's pretty amazing. So this is a plastic heart pretty much, which mimics all the hemodynamical conditions, and the media, of course, is not blood, it's cell culture media. But it works really well. We have done 3 to 4 weeks of conditioning in these conditions. And this was patented as well and has been licensed by a company which is developing bioreactors. So the final step would be, assuming that all of this is perfect, which is not, we still have to work on some of these steps, is to test this in vivo in sheep. So by a large coincidence, we found a very welcoming institution, which is the one where we used to work before in Romania, a medical school, and we applied for grants to the European Union, and we received a total of 3 million euros, which allowed us to put together a large animal facility at that institution and a tissue regeneration lab where we duplicated the bioreactor conditioning, the seeding, the cell culture, and so on. So in the first step, during 2014-2019, there were 70 acellular valves implanted into sheep with very good survival. This is a team of young residents surgeons, anesthesiologists, and so on locally in that medical school. And they chose the right ventricular outflow tract as a site for implantation because of technical ease. And we have pretty good results with those valves. This year, we have started implanting cell-seeded valves. So these are fat collected from each individual sheep, stem cells isolated, differentiated, seeded into acellular valves, and implanted as autologous cell-seeded valves. And they want to go for orthotopical implantation of these valves using open heart surgery. Just a quick example of the valves that were implanted in sheep. Not so great quality echo, but you can see the characteristic clapping effect of the cusps touching each other during closure. More importantly for us, we wanted to know whether this tissue, without any cells, elicits any immune reactions. We couldn't find any. We couldn't find any calcifications. There were no signs of fibrosis and no thrombosis. So that was very encouraging and allowed us to pursue further. Some of the valves we kept for more than a year. This is 12 months. We also had one 14 months. And you can appreciate how supple and thin and non-thrombogenic and non-calcified the leaflets are. This is the implant itself. Histologically, the valve looked pretty well. There's no inflammation, and there was no cell infiltration either. So the take-home message number 3 is that it's possible to generate completely acellular aortic roots. The integrity of the matrix has been maintained using this procedure. Hemodynamics well preserved. Re-cellularization is feasible, but still challenging, so we're working on that right now. And the scaffold itself, the acellular valve per se, are potentially adequate valve replacements by themselves or scaffolds for tissue engineering. So another project that has spun off of this is to generate high-quality acellular valves for implantation without any cells. So ongoing studies, we will continue to use adipose-derived stem cells. Yes?
I'd be curious about knowing when you place the cellularized valves into the sheep, the extent to which new cells have been recruited into the structure that you implanted that weren't originally differentiated by you. So do you have means to try and get at that?
Yeah, we didn't have a lot of samples for this study. What we have seen is that there are some fibroblasts, myofibroblasts infiltrating, and others have done that as well and implanted and explanted. But these cells, you can see them, they stain positive for alpha SMA, but we don't know what they are. And I'm afraid it's a healing process more than anything else. And for this reason, we kind of apprehensive in using these as actual implants because we have no control over what cells might infiltrate and might happen later. And that's why we took the route of re-cellularizing them in vitro before implantation with the right cells in the right positions, trying to avoid that from happening. So there is a larger study done in Germany where they implanted acellular pulmonary valves in pulmonary position. And after a while, they got infiltrated with these cells. They said, good, problem solved. But then if they left them longer, those cells actually reacted like a normal myofibroblast. They fibrosed the tissue and contracted the valve, and the valve became stenotic. It stayed open and wouldn't work anymore. So that's a big problem when you implant these collagen matrices. Typically, they will get infiltrated with myofibroblasts as a healing reaction, pretty much.
Yeah, I guess I wondered whether there might be some sort of slow homeostatic replacement of, say, endothelial cells by other endothelial cells.
Well, these did not endothelialize, really, did not. Yeah. And so that's why we want to actively endothelialize them before implantation. Yes, sir.
Does the cellularized valve have or require any vascularization, or are those tissues, is it sufficient to extract gas exchange and nutrients from being bathed in the...
Yeah. Luckily, all valves are acellular. It's avascular, I'm sorry. Yeah, avascular. Luckily, all valves are avascular, so we don't have to worry about that. And there is a pretty extensive vasculature in the adventitia of the aortic root in the outside. So that's definitely something we need to look into, because reconstituted arteries that don't have adventitial capillaries might not survive for a very long time. That is known from surgery, especially if you clean off the vasculature from the outside. These arteries might die at a later date. So not for the cusp tissue, but for the supporting tissues in the root. So what are we doing now? We're continuously working with adipose-derived stem cells for valve regeneration. We're looking more closely now into taking these cell-seeded valves and adapting them mechanically in the bioreactors before implantation. Why is that? I did not show you data. We did submit freshly seeded valves to implantation and bioreactor conditioning. And a lot of these cells were not alive after 3 days. So we believe that there is a need for a very slow, progressive mechanical adaptation and conditioning in the bioreactor before implantation. And we also want to look closely at ECM homeostasis. We don't have all the tools, but with what we have, we want to look at collagen and GAG synthesis and degradation in these cell-seeded scaffolds. So this would be a very important next project. Meanwhile, since we have the funding for this type of study, we're going to have up to 20 cell-seeded valves implanted in Romania. And hopefully we can measure anatomical growth, because we're implanting these valves in juvenile sheep, and they almost double in size within 1 year. So that would be important, because we believe this is the ultimate test for tissue engineering, having tissues actually growing with the growth of animals. Other topics that I wanted to mention, these are in collaboration with Agneta Simionescu. We decellularize vascular beds, arteries of a variety of diameters. You see here before and after. And we use bioreactors, this time they are vascular bioreactors, to study their properties. And this year, we will actually start implanting these in animals as well as shunts. For myocardial regeneration, which might be of interest to this institute, we have generated either whole hearts, as you see here, before and after decellularization, or whole heart ECM, or just flaps. This is just one individual flap that is connected to its vasculature. And we use these as models to study myocardial pathology in vitro. We also try to recellularize, for example, in this experiment, we have recirculated endothelial cells by perfusing them through the vasculature of this acellular heart. And we found numerous capillaries filled with endothelial and lined up with endothelial cells. So there is a chance that using a whole decellularized heart, we could start to put cells back in, such as endothelial cells in the vasculature. And in these panels, we show that by combining fibroblasts, collagen, and endothelial cells, we could reconstitute the ventricular endocardium as well. So this is an attempt to regenerate whole hearts in vitro. Altogether, as noticed by Rob as well, while doing this, we are both biochemists, so we're not engineers, but the engineers helped us develop platform technologies. We now have technologies to decellularize heart valves, arteries, veins, and other tissues. We have developed some technologies to re-cellularize these tissues, and we have the bioreactor development program as well to condition them. And the vision would be that at some point you would have a system which contains all these components, and you could have off-the-shelf valve scaffolds combined with patient cells, and those would be implanted. But we can also use them as in vitro models for pathology of different tissues. So the program now contains these two components. These are the two labs at Clemson with the bioreactor component, the bioengineering, stem cell biology, biochemistry, and the veterinary medicine, cardiac surgery, and other labs in Romania. With this, I would like to acknowledge this is just a partial list of people contributing to this project at Clemson, as well as the people in Romania. And let's not forget the many, many patients that have been used throughout this experiment, which each one of them has been named with Romanian names. And funding from the NIH, the Dempsey Professorship Endowment, and the two grants that are listed here from the Romanian government. And with this, I would like to thank you for your attention and entertain any questions.
So being a Kiwi, I obviously love to see sheep acknowledged in the last slide. Yeah, go ahead, Mike.
So I can see very much how this is extremely important and potentially useful for treating disease valves. I want to flip the question a little bit. What if you have not a diseased valve, but a diseased heart, say hypertrophic heart, and you create stress forces on the valve, like the aortic valve, that cause it to not be operating properly, and you can't fix with the appropriate surgical or pharmacological intervention the hypertrophic disease. You can replace the valve, but the replacement valve will be subject to the same stresses, I would imagine, the same thing. So I guess the question I'm asking you is, are you thinking about engineering valves with properties to withstand different sets of forces in a diseased heart? It's not replacing a diseased valve that can enable it to work better in that milieu.
Mm-hmm. That's a great question. We're not there yet, but it's a very challenging field to get there. The truth is, with valve pathology, patients that present themselves for valve replacements, typically they will have the stenosed aortic valve, the calcified aortic valve. The underlying myocardium will be reactive. So it will get thickened a little bit, but not to a great extent. I mean, if you really don't look at it at all, you're going to get heart failure eventually. But most of them do not reach that stage of heart failure. And the interesting thing is that after you replace that valve with a mechanical or whatever is now on the market, the myocardial hypertrophy goes back to normal. So we are hoping that we don't have to design a valve for that condition, because that condition will be resolved once the valve starts opening and closing properly. If it's too advanced down the line, then definitely that's a different story. Yeah, I mean, those hearts typically require transplantation. So, Sami.
Right. So I was wondering, like, when you differentiate your cells, so you do that separately with different growth factor, fibroblasts and the endothelial cells.
Yep.
And so when you put them like on the scaffold, that happens also at different time.
Mm-hmm.
But then when both cell types are there, what medium do you use to maintain the scaffold? Do they have like the same differentiation factor for like both lineage?
Mm-hmm. Excellent question. This is one of the questions that we're trying to answer. We don't even know if they would reverse after those factors are being removed. So we will try to find an answer to that soon, because it's important. After differentiation, we do not use differentiation factors anymore. We just use normal media. And that's a great question for anybody using two or more cells. What do? Typically what people do, they use the endothelial cell media combined with the normal media, fibroblast media. And sometimes they do 1-to-1. So your guess is as good as mine. I have no idea what the best media would be. We just kind of combine them 1-to-1. For the bioreactor, this is a pretty big system. It requires about 1 liter. So it becomes very expensive to use specific media. We just use DMEM with FBS and whatever. So yeah, that's a big problem. We don't know even if they would preserve their quality after implantation. So hopefully some of these sheep, after we explant the valves after 6 months, we will want to look for markers and see if it's, the markers were preserved or not. So it's a big question mark. Yeah. Yes, John.
I really enjoyed your talk.
Thank you.
Using the fiber gel, that's something we use in our lab too. And we've got questions about because it's wound healing, it's clotting factors. If that's doing something to the cells to kind of stimulate them down sort of remodeling kind of response rather than more of a physiological homeostatic condition.
Yeah, we haven't looked at that yet. We actually found in the literature quite a few people use it as a temporary scaffold for manipulating cells in 3 dimensions mostly. So we do know that cells make sufficient amount of enzymes to degrade that pretty quickly. So within 3 or 4 days, it's gone. So I'm not saying we're not worried, but yeah, definitely that's something we need to look at.
So they react to it for a short amount of time and then it goes.
Yeah. They secrete plasmin, I think, and in vitro it just degrades very quickly. So
let's imagine that we're 5 years hence, and your valves are going into patients.
okay.
How do you imagine steps as they occur? Does the bioreactor get wheeled into the operating theatre with the valve sort of clicking away on it, or what's the scenario?
The scenario, right now it's not very effective because we require about 3 weeks for stem cell differentiation. So the scenario would be as follows. If a patient can wait a month, okay, he's been diagnosed and is programmed for an intervention a month from now, we collect fat, which is easy, lipectomy, a small piece of fat. Then the fat is sent to a lab. The stem cells are isolated, propagated, differentiated, and seeded onto an off-the-shelf acellular scaffold of the proper size because the patient has been sized up for the right diameter of valve. It's been seeded, conditioned in the bioreactor, and then transported. We don't have a transport bioreactor yet. We're working on that. But then it will be transported to the clinic and implanted into the same patient where the cells were being collected. So about a month. The shorter scenario would be not using any cells, making sure the matrix is appropriate, and just having off-the-shelf, different diameter decellularized valves and ready for implantation. And the secret for that is, I didn't mention here, if you cross-link the matrix, even so slightly, with a chemical agent, it's pretty stable, and it looks like a pretty good alternative to the mechanical valve or the cross-linked tissue valve that is already on the market.
Have you thought about kind of dosing the cells, like sort of, just a homeopathic dose of endothelial cells, fibroblasts, all the way up to, full dose?
We do look at cell density. In tissue engineering, that's a very big problem. Nobody really knows how to do that. You just kind of optimize it based on a trial and error kind of experiment. So we have a pretty good idea of how many cells are needed to cover the entire inside surface, for example.
Well, I guess more specifically, my question is, if acellular works, then how many cells do you need in order to get, the full cellular response?
I don't know that. Yeah. And we need an animal study for that. Yeah. Any other questions? If any of you feel like that dog in the picture in the lab, yeah.
He looks happy.
He looks happy. This is our actual sheep in Romania. This dog it's a stray dog.
Oh, he's stray. I thought it was
Yeah.
a working dog.
He just got trapped in there. So.
All right. Well, thanks again.
Thank you.